Hello, welcome. Today we'll be talking about transcription. Now, the purpose of transcription is to make an RNA strand that will be useful to us by making amino acids, which will then be later used to make proteins, enzymes, and hormones. Now, the first step into transcription is called the initiation process. Now, how this works is a signal or promoter will attach itself to the receptor and bind with the cell. This is a cell. And now what it'll do is transduce or translate down via enzymes, because enzymes will break it down, as we learned from weeks ago. And it will do that until it finally reaches the nucleus. Now in the nucleus we have DNA that contains the genetic material and or what we so it's basic genetic material is basically the blueprints we need to build the amino acids. So, and without these blueprints you can't really build anything. It's like having a brick and not knowing what to do with it. Yeah, it's like that. Yeah. And let me open up my other picture. And this is where it really begins. Okay. So the signal comes and attaches itself here at the TATA box, where many adenines and thymines are attached or grouped up together, and it will attach here at the prime 3 strand of the DNA, as seen here. And the main role that the RNA polymerase does in transcription is to make an RNA strand that will be sent to the ribosomes, and that's what I'll talk about later. Okay, so it arrives specifically to the chromosome and DNA it needs to build the specific codon the signal needs, the amino acids we need. Okay, and this RNA polymerase will travel from a prime 3 end to a prime 5 end. Let me just clean this up. Yeah. So, yeah, it'll travel from 3 to a 5. And an enzyme called helicase splits this DNA strand into half. Unlike here, how it's bonded, it'll split it in half, and that'll allow the RNA polymerase to read the codes it needs to build the RNA strand. Now, so it reads opposite, and what I mean by that is, if it reads a, it will build a U. If it reads a C, it will build a G, like as shown here. Like how here it has a U, a C, a G, an A, a U, a U, a U, and whatever. And those are now called codons. Or that's the code that's gonna be needed to make the amino acids. And you'll keep doing this until it finally reaches the stop codon that tells the RNA polymerase to stop reading. Now, while it's moving this way, it will build an RNA strand through the back. Yeah. So, now, let me just clean this up real quick. So, now that has happened, we have an RNA strand right here. And with that, we can now take it and transport it to the ribosomes. And in the ribosomes, that's where we have all the proteins made. That's where all, as we learned in 9th grade, that the ribosomes create all the proteins. And before going on, we will have to put a 5 cap. And basically what a 5 cap is we put it here on the RNA strand. Basically, what it is it's like protection in case if the RNA strand gets destroyed. So yeah, and out the back we will have a poly A tail out the back. So yeah, and now it will leave the DNA strand, and the DNA strand will now close and bond together again by the base pairs, because we don't need it anymore. And the reason why is because the whole reason why we make an RNA strand and not have the DNA strand go into the ribosomes is because if the DNA strand gets destroyed or messed with, like, this will create huge problems in our body, and we can't afford to lose our only DNA. So now that we have the RNA strand, we have to skim through it, and since all these codes are, we still have a lot of codes, we need to skim through it and see which ones, see which codes we need and we don't need. Now, the codes we do need are called exons. And now, hold up. Exons. These are exons. Now, exons we need because those are the important parts, and that's the stuff we need, or the codes we need to build the amino acids. Now, there's also stuff we don't need. Oops, same color. Called introns. That we don't need and is not important and will stay in the nucleus because we don't need it for the amino acid. So this will be skimmed through, and while it's being skimmed through, the exons will remain in this the RNA strand, and the introns will be taken out. Yeah, this is a process called splicing, and after this we will end up with a finished RNA strand called mRNA, or also known as messenger RNA. So before the splicing process, we had mRNA. I mean pre-mRNA. Now we have mRNA, and that RNA will be transferred to the ribosomes, where translation, our next process, will begin. So termination processing was when the mRNA needed to get the ribosomes. Yeah. Thank you.